you know that brain is the central organ, but the central organ cannot go to every place and work like as in the case of blood. Blood itself goes and carries out the functions. It cannot be in the case of central organ, that is central regulatory organ, which is brain. So brain has neurons and the nerves which carry the information, which carry the signal and take back the signal in the form of motor signals and sensory signals. We are very well aware of that. Now what we have to understand is that how that impulse which carries the signal that is being generated and carried forward from one neuron to another neuron. So for that we need to see the structure of nerve cells. We are quite aware of the structure of nerve cells by now, that there is an axon, there is a cell body, there are dendrites which are in close connection with each other. Now what are the characteristic of the nervous cell is that these are excitable cells. Now when I say excitable cells, it refers to the membranes which are present in them which are polarized. Now another term that is polarized, polarized means that supposedly this is the axon I am talking about, let alone this would be cell body and this would be synaptic region. This axon part, this axonal membrane would be sort of polarized. How it would be polarized? That there would be a potential difference between the outer membrane and the inner part. That is, the outside of the membrane would have a different potential and the inside would have a different potential, and there would be a potential difference which would be present between the two because of the different ionic concentration that are present across the axonal membrane. Now what supposedly there we are having more concentration of sodium ions and inside we are having more concentration of potassium ions. So this concentration would vary, and due to which the charge would vary and due to which the potential difference would be varied, and the difference that would be there. Supposedly I have minus 40 millivolt and over here I have minus 70 millivolts. Just for example I am taking, don't get into depth of these values. There is a difference and the membrane would be believed to be polarized because the difference is present. Now apart from these, the nerve cells also have voltage gated channels. Now what voltage gated channels means? There would be certain proteins. You know, we are talking about a membrane of a cell, that would be a plasma membrane basically. There would be proteins which would be present. We have the structure of plasma membrane in our mind and we remember that there are porin proteins which are present, or the membrane proteins, transmembrane proteins which enable the transfer of ions through them. So those proteins, they act as gates. Normally a gate is a regulatory part of the house which acts as a checkpoint whosoever has to enter a particular place. Supposedly we are talking about a gate of a park. At that gate there would be a decision which would be made that who has to enter and who is not allowed to enter. Same way we have these voltage gated channels. Now these gated channels, they are more dependent on the voltage. That means they are electrogenic basically. They would be opening up depending upon the polarization state of the axon. So the next point is that what is the basic tendency of the exolemma, that is the membrane of the axon, and these voltage gated channels. It is that in the resting stage, that means when the membrane is polarized, when the impulse has not arrived. So we have another term, that is resting stage of the axon. In the resting stage, the permeability favors more towards the potassium ions as compared to the sodium ions. That means the sodium ions, they are not able to move inside the axon as compared to the potassium ions. Potassium ions are more permeable, the membrane is more permeable for potassium ions as compared to the sodium ions. So what happens is that through these gated channel, voltage gated channels which are driven by ATP, there is movement of 3 sodium which go out and 2 potassium which come inside the membrane. This is ATP driven electrogenic pump I am talking about, not about the permeability. You see over here, the permeability is towards the potassium ions getting inside. The potassium ions are otherwise more permeable, more favorable towards the exolemma in the resting stage, which gets altered when the resting stage is changed, it comes to the excitation stage, then what happens is due to the voltage gated channel, sodium permeability changes. We are going to come to that later on, but for now you are going to concentrate more on the resting stage, that in the resting stage the membrane itself is more permeable to potassium, it is not permeable to sodium. So the potassium they come inside, the sodium they remain in outside. At the same time there is one thing, that is the organic acids which are present inside the exolemma, which are present inside the axon's exoplasm, they are negative in charge. So what happens in the resting stage, the polarized condition, outer exolemma is positively charged and the inner exoplasm is negatively charged. So this is what is the resting stage of axon basically is all about. Now at this resting stage there would be an excitation which would come. That excitation would enable the ATP pumps to open up, and those ATP pumps, they allow 3 sodium outside and 2 potassium inside, due to which this positive charge which was outside, the polarity was positive and inside it was negative, it gets altered. It keeps on moving forward, that we are going to understand in the conduction of nerve impulse. But for now you need to know that in the resting stage, that is when the axon has not received, or the nerve cell has not received any sort of input or excitation, it would be having an exolemma which would be more permeable to potassium as compared to sodium. It would be having a charge that would be positive outside due to presence of sodium ions and negative inside due to presence of lesser potassium and more of negative organic acids which are present inside the exoplasm. And then after understanding this resting stage, now we have to see that how the conduction of impulse takes place. First of all, there is an initiation of impulse, and that impulse is by excitation. There are excitable electrogenic pumps which alter the charge of the exoplasm, that is known as initiation of impulse. Once that charge change has taken place, there would be transmission of impulse along the nerve fiber. It would move further and further and further, like in the same way the current passes. You switch on the light at one place and the tube light lights over somewhere else. So that is basically the flow of charge that has taken place from one point to other, or as in the simplest case you can realize that supposedly you put a spoon on fire, it would slowly and steadily come to the handle, the heat would come, you would feel the heat in the handle because it has moved further, it has transmitted further. In the same way, the charge change that has occurred, the alteration of charge that has occurred, that also gets transmitted, and that is known as transmission of the nerve impulse across the nerve fiber one by one by one. Okay, then the second stage is when the synapses come, when this neuron ends, the transfer of the impulse has to go to target nerve or the target muscle, and then the target muscle has to work, the target tissue has to work accordingly, whatever signal has been brought about. And that signal again won't be a written letter, supposedly it would be in terms of the potential difference only that would be perceived by the target organ. Supposedly we are talking about the target tissue, which is a muscular tissue, as in the case of neuromuscular junction. So we are going to revise. What happens is that there is an electrical excitation that has to come to the nerve cell, and the characteristics of the nerve cells are very much suitable for the electrical excitation that comes over there and move further and further and further. And that further movement is due to the presence of the characteristics that we have mentioned over here. There is a potential difference which occurs in the membrane. The membrane is polarized and the concentration of sodium and potassium varies, and the concentration varies on account of the permeability differences. As soon as the excitation reaches, that permeability changes. There are ATP generated pumps which work opposite and the polarity is reversed. When the polarity is reversed, now over here you have positive negative. In close contact you have negative positive. So charge is bound to move. There is a closed circuit formation that takes place. please recall your knowledge from physics, how the closed circuits are formed, positive, negative, negative, positive. A circuit is formed and charge moves further. Then again a circuit is formed and charge move further, and this takes lesser than 0.3 millisecond, because whatever I am speaking, whatever movement I am doing, this all is taking so less time, and it is all because of the nerve impulses which are starting from the brain, they are coming to the muscles, and from there they are perceived. So this is what all I have written. Supposedly I have to move this hand and show you like this. So this has occurred like this. An impulse was generated in my brain, it is being transmitted along the nerve fiber, it came to my hand, target muscle, and from there I moved that target muscle. This is what we are talking about, the conduction of nerve impulse, how the positive negative charge variation and the polarity variation is able to move the body and is able to is enabling the body itself to carry all the functions, which are very baffling to understand that how it takes place. Next we are going to understand in the next lesson what is the basic principle, what is an action potential, how does the nerve impulse go further and further, and for your knowledge, one thing you have to remember, the last thing in this lesson is that an oscilloscope is used to measure the differences or the potentials which are present in the nerve fiber, and minus 70 millivolt is supposed to be the resting potential of a neuron. Now you might ask me that why minus 70? Minus 70 because we are referring the inside of exoplasm, and the inside of exoplasm we know that it is negatively charged due to organic acid. So this is the resting potential of the membrane that we are having when the excitation has not come. Next we will see what happens when the excitation comes and how it is moved further.